This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 440, recorded on May 5th, 2017. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And it is, actually, I forgot to bring up the weather report before we started, but yeah, it's pretty much the way it's been all day. It's cloudy and rainy and foggy and 51 Fahrenheit, 11 C. Yuck.
Yeah, it's 14 here. We also had this big storm this morning.
Yeah.
Rain pattering against the windows. Yeah. We missed the 4th yesterday, right? May the 4th be
May the 4th, that's right.
Also, but that means today is Cinco de Mayo.
Cinco de Mayo, that's right. Also joining us from southeastern Michigan, Kathy Spindler.
I couldn't wait to be introduced.
Yeah, it's okay. There's no rule.
That so, kind of is an informal folk way that we have, but yeah.
Right. It's 46 degrees here, raining. It means it's 8 degrees Celsius. But by Sunday, it's going to be sunny, and that's important because that's when the Waterhill Music Festival is. That's the music festival that's in my neighborhood. Oh. People play on their front porches between 2 and 6 in a highly coordinated schedule. It's really fun.
Nice.
Neat.
How was the choir last week?
It was really good. We had a great performance.
Wonderful. Yeah. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
I'm well, and you? You've been well? You have a good week?
Yep. Everything's good. We've got it's 81 degrees, clear blue skies. It's beautiful. That's 27 C, I think I looked it up. 27.
27, yeah.
So
82 would be 28, right, Kat?
This is, I'm told, because I'm still learning about Austin here, I'm told unseasonably cool for this time of year. At 81 degrees. It was like 57 this morning or something like that. But it's a beautiful day. I'll take it. This is good.
Well, this week, this is the last week of classes this past week. Monday was the last day, so my virology course is over. It's study week at Columbia, and Monday is our final. So
wow, they get a week. Our students get a day.
Really?
Yes.
That's funny.
Yes.
Wow, that's not very much.
No.
Nope. Wow.
Gotta
well, anyway,
stay on top of things.
I actually have 5 students in the lab taking it today because they had a conflict on Monday, so they're working away. Hopefully they don't have any questions for me. It's a good exam, 8 questions, 3 hours, should be a piece of cake. But there'll be a curve, of course, as there always is. Did I say that Dickson was away? I don't remember.
You said, I don't know.
I think you said that before we started recording.
So Dickson is away for the next month. He won't be back till June.
He's fishing in South Africa?
Yeah, I think he's going to Milan first for some indoor farming thing, and then he's going to South Africa.
Probably a fashion show. And
I want to tell you about the American Society for Microbiology Microbe 2017. And this takes place from June 1st through the 5th in New Orleans, which is in Louisiana. Will provide a unique platform to explore the full scope of the microbial sciences at ASM Microbe 2017. Will feature an exciting program with opportunities for you to gain great new science and connections. Whether you're a researcher or educator, a clinical or applied microbiologist, an infectious disease or environmental microbiology expert, explore the complete spectrum of microbiology at ASM Microbe 2017. For more information, asm.org/microbe. We will be doing a TWiV and a TWIM from ASM Microbe. Rich Condit will be joining us for the TWiV, and the keynote speaker for ASM Microbe is going to be Kate Rubens, who is a virologist turned astronaut.
Right.
She has been up in space. She's the person who sequenced DNA up there.
Yeah, we tried to get a hold of her because we wanted her to come to ASV.
Mm-hmm.
And there was no way we could get through the firewall of NASA to email her. I tried about 5 different ways. And then eventually we found out that maybe Paul Dupree had a connection. But I wanted to get her before ASM got her, but obviously we failed.
Well, I have the PR person you need to contact. I have her email. So if you want to try next year, I can give you that. All right. Speaking of the American Society for Virology, that's coming up too, June 24th through the 28th in Madison, Wisconsin at the Monona Terrace. Early bird registration ends midnight Central Time, Friday, May 19th. And after that, the registration fees go up, so don't be late. ASV.org, and you'll find a link there, right, Kathy?
Exactly.
We have a bunch of follow-up because we didn't want to use up Paul B. Nash's time last time with it. From Maureen, hi, my instructors in science. I just listened to TWiV 438 and have to say I agree with Bob's letter. I have been meaning to write a similar letter asking that parts of episodes be addressed to those not as well versed in the nuances of virology or microbiology. I love and have listened to all the TWiVs for years, but I do have a wish sometimes that there could be a summary for those of us who need it. I was thinking that a short layman's explanation before the paper is read, e.g., this paper is important because blank and it shows blank. We're excited about it because blank. After discussing the paper, a few sentences that say, in summary, the authors wanted to explore blank and the conclusion of the study was blank, especially the TWiM episodes. I really enjoy listening to the episodes, even though I've
read some studies whose conclusion was blank.
Yeah, that's right. And what about, we're excited about it because... Well,
blank.
Yeah.
Because, you know, the thing is, when papers get on TWiV, we're usually excited about them.
Yes.
We don't talk about non-exciting papers. So we'll tell you why it's excited. Let's see. I really enjoy listening to the episodes, even though I haven't a clue what you are talking about sometimes, but I just wish I had a little clue. I like that. Anyway, I agree, too, with you that the conversation mode, rather than the lecture mode, teaches us well and makes us feel included. I often fall asleep listening to your podcast and find my mind immersed in your conversations in my dream. However, I keep wanting to join the conversation in my dream because I become someone important, and none of you will shut up to let me say something. I must remember to put my timer on to limit my listening while falling asleep. That's funny. It's the same thing Paul B. Nash said, right?
Yeah.
Right.
He said you can't get a word in. But it's very hard, even if you're on the show, to get a word in.
Yes, Kathy probably has that dream a lot.
I don't dream about it, but I do when I listen to when I do listen to TWiV or TWIP at night, I do set the timer on my phone so that I don't have to rewind quite so far.
Really?
Yes.
How long do you do it for?
Usually 15 minutes.
Interesting. Do you think you're doing any subliminal listening? Do you remember anything the next day at all?
Well, it's hard to know, but
Yeah.
I don't think so.
Interesting. Yeah, we had a listener at Berkeley. I don't know if she's still listening, but she said she used to fall asleep. She actually said, I like falling asleep to the voices. It's really soothing. Well,
it's that good sound quality.
Yeah. Okay.
At any rate, Maureen is absolutely right, and we should do this.
We'll try that. We've already started on, last week Paul gave a good summary, but we'll do one up front, and I started it on TWiP also. So once we bake it into our routine, then we won't forget, right?
Right.
Yep.
Alan, can you take the next one, please?
Sure. Niraj writes, Dear all, this is Niraj, and I'm mailing after a long time on TWiV, but not because I've not been listening. That never happens. But because I've been distracted with a lot of things, and in between the chaos, there's always room for TWiV.
Chaos.
I'm particularly intrigued by the last couple of episodes. From episode 437, I was astounded to hear that getting repeatedly vaccinated against seasonal flu isn't that beneficial. Could that partly explain the poor cure rates for some of the recent vaccines that have been developed? In the same light, the discussion highlighted the importance of vaccinating young children. Someday I shall explain to my 2-year-old the benefit of that needle prick which made him scream from the deep recesses of his lungs. Yes, the immune system has limited capacity, but do we really understand the reasoning behind why doesn't our body develop as strong a memory response against subsequent viral challenges as it does to the first one? Is it the nature of the virus or the differences differences so subtle that our surveillance fails to detect and magnify it. Fascinating stuff, and just goes to show that even after decades of research, we're still learning things about such a small yet powerful entity, i.e. the flu virus. Given the current breadth of research, I'm hopeful that someday we will completely move away from generating these viral strains in chicken eggs and develop an orthogonal way of overcoming this situation, or overcoming this infection. Good use of orthogonal. We can probably already do. Let's just tackle these one at a time. So getting repeatedly vaccinated against seasonal flu protects you to some extent from that season's strains, assuming there's a good match between the vaccine and the strains that are circulating. But it doesn't really help you much if you have different strains circulating the following year. And yes, that's the core problem in flu vaccination. And no, we don't understand how most of that works.
There is some interesting work published recently about how the first flu infection you get, whether it's a natural infection or a vaccine, really determines how you're going to respond after that. And I'm going to do that paper in the next few weeks. So stay tuned.
Okay, good. We did a paper long ago on original antigenic sin, I think.
Right.
That's right. Same idea. Yeah.
The same idea, right? So it would be good to refresh that.
In fact, in this paper, they did extensive epidemiology, serology to look at that, and the results are very interesting. So, with a lot of data.
Sounds like a great TWiV paper.
Yeah.
All right, continuing with Niraj's letter. On a more subjective note, I really liked the March for Science discussion as part of TWiV 438. I do agree that telling stories about science is far more effective than presenting cold, hard evidence only. Evidence can definitely be presented in support of the discovery, but one always remembers and appreciates more if one knows the origins and details of the journey that was undertaken for the discovery. I can personally share an experience from my graduate school days at Rockefeller University. I was admitted to the TPCB program, and in the initial stages of the program, there was a provision wherein the students had a chance to meet with the participating faculty one at a time for lunch and introductions. A lot of the professors presented data to highlight what they were doing, but the interaction that captured my imagination was the lifetime chance to meet with Dr. Paul Nurse, Nobel laureate and the university president at the time. He didn't show any slides, but shared his passion for science and how it helped him to discover cyclins serendipitously in yeast. The whole episode is still vividly registered in my head, and I will never forget the humility and eagerness with which he urged us to attack the problems we care about with full vigor. Failure happens, it's the nature of science, but it's still a data point, and at times a very important one. It's always harder to know what not to do than to go do something. Like Thomas Edison famously said when asked about the failures he undertook before inventing the light bulb, he said, "I didn't fail, I discovered 20,000 ways of how not to make a light bulb." Attitude is a big component of success in general, but especially so in science, given that the majority of things fail. In the present scenario, I feel at times scientists get too sucked up by the data. The nature of the trade has also made it harder to work on things where there's no foreseeable therapeutic benefit. I'll never understand why. Science communication is tough, but is a necessity one should engage in. It doesn't have to necessarily start at the level of society or have to be big, but it has to be simple to understand and easy to remember, especially if one can mention the impact it has on our understanding of something. Overall, I do feel there's more noise than order when it comes to the way the science gets publicized, and with the current administration hell-bent on doing deeper cuts, I hope not, I worry about how one's enthusiasm can stay afloat to pursue a career in it. Someone once mentioned to me, ideas are cheap, experiments are expensive. I don't think I was in agreement then, and I don't think I am completely in agreement now, but I do think there's a fair balance that needs to be struck. In the end, I apologize for the lengthy rambling, but I would like to sincerely thank the TWiV series of podcasts for making many of us more literate about the diversity surrounding us with an honest dose of rationality. These podcasts are much more than just scientific discussion. They're a tool for shaping how one should think about discussing science. At least that's what I feel and experience. March on. And Niraj is a scientist at Sutrovax. Thank you very much.
Yeah, excellent. I like the, is this a male or a female?
Guy. I think it's a guy.
I like the way Niraj says that the podcasts are a tool for shaping how one should think about discussing science, because that to me is the important part of this.
It's an Indian masculine name.
Okay.
I looked it up. There's this thing where you could look things up.
Oh yeah, I've heard of that.
It's
called a library,
interweb,
right?
I just went down to the library. I came back really quickly. So this whole idea of facts and data, ever since I read the article by Tim Reckwarth, which we talked about 2 weeks ago on the Science March TWiV,
right?
I'm afraid of giving people too many facts here on TWiV. I don't want to drown people in them. So we have to strike a balance because I think sometimes we do. Sometimes we get into the weeds clearly, but I think we have to be careful, right?
I think Niraj makes another good point that not all science communication has to be aimed at everybody. And I think we have a kind of a niche that we've carved out for people who want to hear scientists talking as scientists. And yeah, we get into the weeds because sometimes that's what you do, and that's how fields progress, and that's how we discuss these ideas. But yes, it's important to have some balance where we back up and say, the reason we're going into these weeds is because of this
that speaks to the previous letter. As long as we come up for air now and then and say, okay, this is what's happening here.
By the way, we're going to have Tim Reckwarth on TWiV in a couple of weeks, so he'll come and we can talk about him and what we're doing. And also, I think we haven't found all those people who like to hear scientists talk yet.
Oh no,
I think we still. And
I think there are a lot of people who would like to hear scientists talk, but they don't know it yet.
Yeah.
This idea about failure being good, I think I mentioned this on a previous TWiV, but I'm going to do it again. Reminds me of this thing I heard Neil deGrasse Tyson on Wait, Don't Tell Me. Mm-hmm. And it's one of these things where you have to answer 3 questions or something like that, and you'll get somebody's voice on your answering machine, whatever. At any rate. So apparently he got 2 out of the 3 questions wrong. And at the end they said, oh, geez, I'm really sorry that happened. He said, what do you mean you're sorry? He said, I learned 2 new things I didn't know before.
Yeah.
Yep. Kathy, can you take the next one?
Sure. Laurel writes, Dear TWiV team, good afternoon. This is my first time writing, but rest assured I am a long-time listener. For the past 5 years or so, I've enjoyed listening to you all, whether it's been during my marathon training while using the microscope to study human papillomavirus in Paul Lambert's lab at UW-Madison, or while dissecting fruit flies in Haifan Lin's lab at Yale. Our weather this afternoon here in New Haven, Connecticut is gray, humid, and approximately 18 degrees Celsius. It's similar to the weather at the March for Science, so I think it's appropriate to share a few reflections from last week's TWiV episode 438 on science communication. Before I heard from the entire TWiV crew last week, including Kathy Spindler at our Yale Science Journalism Symposium, thanks again, I had a limited view of science communication. I thought it was solely about translating the complex language of science into accessible information. I still think that this is an important aspect of science communication, especially in the classroom, but I now appreciate the importance to connect science with people and their beliefs. I see how relevant this connection is, particularly when I reflect on my beliefs and evidence of climate change. Today, I accept that humans have rapidly changed the climate of the Earth. Yet for many years, I tried to deny and ignore the evidence. It was easier to deny the evidence than to accept that I have contributed to global climate change. As a scientist, I believe that if we want to use evidence to change policy, such as climate change, vaccinations, and education, then we need to engage in conversations that incorporate both beliefs and evidence. Relatedly, one of last week's TWiV audience members, the microbiology professor, asked about tools that the scientific community can use to teach students how to critically analyze what they read, especially on the internet. One tool that I would like to suggest is Sally Hoskin's CREATE program. In the CREATE program, which I'd like to nominate for a listener pick, students analyze articles from the internet or printed sources such as Wired, New York Times, or National Geographic. Through questioning what they read and the experiments that would be required to reach the article's conclusion, students become critical of how data is interpreted and portrayed. While at the University of Wisconsin, I really enjoyed implementing the CREATE program in a freshman biology course. Another highlight of the class was that the students also had the chance to meet scientists, including Shelby and David O'Connor. Speaking of David O'Connor, a reoccurring guest on TWiV, some of my favorite TWiV episodes include those where there is a panel of scientists and we hear their stories of how they became scientists. Thanks for creating TWiV. I really enjoy listening. All the best, Laurel. P.S. You can read about my own story of how I became a scientist on my blog here. And she included the link to her blog, which I went and read, having met Laurel, of course, in person at Yale. So it's a good blog.
You know, it's interesting that she brings up this guest issue. So I notice when we do on-the-road episodes, they get fewer downloads than the normal ones. That's a pattern. Now, it's not just a guest, because last week, Paul Benash actually was higher than usual that first download day. But I noticed that the David O'Connor one, when I was in Wisconsin, it gets fewer downloads. The one in Washington got fewer downloads than normal. I don't know why that is. First of all, people should be subscribing so that you automatically get downloads. And that way you keep our numbers up, even if you don't listen. And then, why would you not download it? You look at it and go, I don't want to hear a review. Probably the sound is not good. I don't know why it's less. I'm curious as to what's going on.
Yeah, but Laurel's got just the opposite.
Yeah.
She likes those. So
she did.
It
I'm
could
sure
be
there's
like people with the weather. There's some that like it and some who don't.
Yeah.
But I would. That's really, really weird, because those also tend to be shorter episodes.
Yeah.
And I would kind of expect people to gravitate to those. Oh, do I want to get into this 2-hour? Oh, here's a 1-hour episode. That's really odd.
Don't know. Rich, can you take the next one, please?
Sure. Excuse me, I got to pan back. I was goofing off.
Hey.
All right. Neil writes, Dear TWiVcasters, yesterday I finished listening to episode 438 on scientific communication. This episode also includes a letter pertaining to the origins of the use of the word bug to refer to all insects or to bacteria. Last December, I published a commentary attached and directly related to scientific communication on the use of the word bug as modified by the prefix super to refer to bacteria exhibiting resistance to multiple antibiotics in pathogens and immunity. My thesis is that the routine reference to bugs that are super when discussing bacterial pathogens with resistance to many drugs is frequently based on minimal thought. Furthermore, such intellectual laziness leads to an overly narrow perspective on the critical challenge of controlling the spread of resistance mechanisms. Regardless of what you think about my perspective, I believe the topic is worthy of a broader discussion. By the way, Pathogens and Immunity is a relatively new journal hosted at Case Western Reserve University. The founding editor, Michael Letterman, a well-known expert on HIV, and the other senior editors, of which I am one, are dedicated to improving the experience of investigators in submitting manuscripts. So, for example, we permit submissions in any National Library of Medicine approved format and only require adoption of our preferred format after acceptance. Anyone interested in additional information on P&I should visit PAIjournal.com. With respect to the claims that several of you made about science exemplifying the pursuit of truth and the reliance on evidence and logic, science at its best does certainly approach these ideals. Of course, science is not always at its best, as many of you have noted at various times. Some scientists, clinician investigators, and science journalists make dubious claims and these examples of exaggeration are by no means rare. The attached brief essay explores the sources, science-related and from other arenas, of what I claim is an ever-increasing volume of statements that are proffered primarily for motives other than promotion of the truth, i.e. what can be described as BS. Since this essay was published, I have settled on a new term to denote the totality of BS, which was inspired by the revolution in biomedical methods for integrating thousands of genes, transcripts, genes, products, and metabolites in parallel. Thus, all BS can be referred to as the bullome. The field devoted to studying the sources and nature of BS, an aspect of the study of cognition and critical thinking, can therefore be called bullomics. I love it. That's good.
And I
Best
believe
regards. Go ahead.
Rich, didn't you used to have the founding textbook on bullomics in your office?
Yes, it's still around here somewhere. I used to keep it out to scare students and people interviewing. It was given to me by my son-in-law, and it's called
On BS.
Yeah, On BS. That's right.
Yes.
That's right. Written by a philosopher, as I recall. And of course, it's a delightful book because it actually is a good treatise on BS, but it's somewhat tongue-in-cheek. So it's ironic that it's BS itself.
Yeah.
Hey, right, this is Neil Greenspan, who's a professor of pathology at Case Western.
Maybe, Neil, you want to come on and talk about the bug issue and the Bolome.
And Bolomics.
Bolome.
Yeah.
So I forgot to attach the papers, but he sent one in Pathogens and Immunity, which you can go to. I'll put the link in. It's called Infectious Super Carelessness in Discussing Antibiotic-Resistant Bacteria. And then another one published in Philosophy Now called The Ever-Expanding Kingdom of Bull. Neil Greenspan takes an appalled glance at all the BS in academia and beyond. I didn't know that Harry Frankfurt had published something called On BS, where it was actually spelled out.
Yeah, I'm gonna take a little break here and go get this. I see where it is.
You're going to go get the Bolome? Well, speaking of publishing, I think we didn't mention that bioRxiv got a bolus of money from the Chan Zuckerberg initiative, right? We didn't say that, did we?
No, we didn't. I did see that there something like that happened. Yeah.
So Chan Zuckerberg has this initiative which gives away money because the Zuckerbergs are very wealthy, of course. And they gave some money, and how much they didn't reveal, but which will allow staff payment for staff, technology development, and infrastructure at bioRxiv, which, of course, published its 10,000th manuscript very recently, and that's housed at Cold Spring Harbor. So that's good. That's good that they'll have money to float for a while.
Yeah.
Very good job. I like that. Rich Condit.
This book is a riot. So Harry Frankfurt is a renowned moral philosopher, professor of philosophy emeritus at Princeton, and cites his books. And I'll send you a picture of the cover and a picture of the first page. I can't read it because it's a
family show.
I can abbreviate it.
Yeah.
The first sentence is it. One of the most salient features of our culture is that there is so much BS.
Yep.
And it goes
It's true.
on from there.
It's always been true. All right, we have a brief note from Jeremy Luban, who writes about TWiV 439. Great podcast, guys. Clear, articulate, interesting, exciting. The PERV CRISPR target number is 62, porcine endogenous retroviruses. Never underestimate George Church. Now, last week I had mentioned to Paul B. Nash that I thought that someone had knocked out all the PERVs in a porcine cell line for transplantation purposes. He said, I don't think all of them, maybe just a few. And no, it was 62. George Church is unbelievable. And
wow.
Then just today, Dennis wrote, "Hi, docs. In TWiV 439 with Paul B. Nash, wonderful TWiV, Dr. Vincent raised the idea that someone is trying to remove many porcine viruses. George Church, in a talk, described how they are trying to remove 61 viruses simultaneously using CRISPR. If this can be done, then porcine organs can be used for human organ transplants. I don't know the latest. That's exciting work. His talk can be found on YouTube, but I haven't searched for the link. On mobile right now. I think it was a World Science Festival panel discussion with several biochemists. Best to you, and thanks for the education." Yeah, so I guess it's done because there's a paper. Jeremy sent the link to a paper, Genome-Wide Inactivation of Porcine Endogenous Retroviruses.
And who doesn't like CRISPR pork?
Ooh. So we do that paper, we can use that for a title.
Yes.
All right, we're going to do the paper just for that.
Just for the title.
As a snippet. A snippet
of CRISPR pork.
Excellent. We do have a paper for you today. And this was sent to me actually just before the ASM TWiV a couple weeks ago. And I had it lined up in case our TWiV fell through for whatever reason, but it didn't. And it was Jean-Michel Claverie had written to me, "I know how much you are into the saga of giant viruses. You will find enclosed my translation of a press release from the CNRS concerning a forthcoming publication in Nature Communications. I also enclose a copy of the proofs of the article. I trust you to respect the strict embargo imposed by nature. Best regards and happy Easter.
We always obey the laws of nature.
Isn't it just an embargo? Why is it a strict embargo? What is the difference, Alan?
I don't think there's a difference.
I mean,
I have not heard of a loose embargo.
Loose. What would be the point, right?
Yes.
And of course, Jean-Michel Claverie and Chantal Abergel are two investigators who have been on TWiV. They work on giant virus. They are at Aix-Marseille University in Marseille, of course. In the paper, we will tell you all about.
Which is now past the embargo date, so we're no longer strictly prevented from talking about it.
That's right. It's been published, and that's called noumeavirus. Noumeavirus replication relies on a transient remote control of the host nucleus, and the authors are Fabre, Jeudi, Santini, Legendre, Trochasek, Coutet, Jean-Michel Claverie, and Chantal Abergel. The first two authors are co-first authors. And this work comes not only from Aix-Marseille University, but the Université Grenoble-Alpes, the Commissariat à l'énergie atomique, INSERM, the Hôpitaux de Marseille. And I just mentioned all of them because I like to pronounce the word.
Because you like to pronounce them, yes.
I do like Italian, but even French, which I took for so many years in my life, I must remember something. This is all about a giant virus, and it's all about a particular family of giant viruses called the Marseilleviridae, which is a family with 5 lineages, and these of course infect amoeba, acanthamoeba. And there are 10 members of the Marseilleviridae. If you would like to be a member, just write us an email. We can do something about it.
We'll have to sequence you, though, and stick you on a phylogenetic tree.
Big.
So here's the summary of this article before we start. There are some DNA viruses that go in, their genome goes in the nucleus, and it's transcribed and replicated there, and many depend on host enzymes to do that. There are other viruses like the famous Condit pox viruses, which replicate in the cytoplasm. And as Condit once told me in this wonderful phrase, he said, pox viruses don't deal with the nuclear bureaucracy. They're out in the cytoplasm. And they have their own enzymes for transcription and replication. And the one here that they discovered seems to replicate in the cytoplasm, but it doesn't have everything it needs to get started. So in the first hour or so of infection, it somehow manages to pull things out of the nucleus and into the cytoplasm and then get things going. And then the flow stops and the nucleus is left alone. So this is transient remote control.
I have my own take on the top-level summary of this as well. So this is about the bootstrapping problem. If you think about a computer, the operating system controls what programs are running and handles pretty much everything. But how do you start the operating system? Because the operating system is what you use to start everything. So on the computer, there's a bootloading sequence that automatically runs as soon as you turn on the computer, and that's bootstrapping. That's why you call it booting the computer. Viruses have a similar problem, especially DNA viruses. Now, RNA viruses, especially a positive-stranded RNA virus, is very, very simple. You get your genome into the cytoplasm, and in many cases, the genome is a messenger RNA. It just gets translated, and then you've got your proteins that are going to carry out the viral life cycle. So that's great. But if you're a DNA virus, you have a bigger problem because the machinery of the cell that deals with DNA is locked up inside an additional membrane inside the cell in eukaryotic cells in the nucleus. So there are two main solutions to deal with this, and they're the two that Vincent just mentioned. You can either say to heck with the nuclear bureaucracy, we're going to do everything in the cytoplasm, but in order to do that, the virus particle actually has to pack some of the proteins that it needs to get started. So in addition to the genome, it needs the proteins that are going to transcribe at least a starter set of genes to get the whole process going. The other option is to do what herpes viruses do and actually make an additional entry into the nucleus and then go and do your business in the nucleus. And today we're going to talk about an example from the giant viruses that doesn't do either of those. And it's kind of cool that it's got this kind of halfway solution that still allows it to bootstrap.
And the other thing about this paper that at least the authors are excited about is that these all have, in their minds at least, implications for the evolution of viruses.
Yes.
So whether you started with a cell and threw stuff away and wound up with a virus, or whether you started with a small replicating piece of DNA in a cell and then added stuff to make a virus. And to them, this is sort of an intermediate in that process that may speak to which of those evolutionary pathways might have happened.
All right. And for those of you that's enough, you can go back to whatever you were doing if you want to hear the details.
Turn on your sleep timer.
So this virus was found in a muddy sample of fresh water from a pond near Noumea Airport in New Caledonia, and hence the name noumeavirus. And they grew it up on acanthamoeba cells, and looking at the infected cells saw icosahedral particles 200 nanometers in diameter, and this is a feature of Marseille viridae, 200 nanometer particles. They sequenced it, of course. Genome of double-stranded DNA, 376,207 base pairs in length. And I don't tell you that because I want to give you a fact, but it's what it is. It's exactly
and that's
that.
Kind of, as giant viruses go, that's on the smaller end of the giant viruses.
It is. Mm-hmm. Now, Rich, what's the biggest giant virus genome we know of?
Oh, I don't even know. I'm thinking like 1.5 megabases or something, or are they?
Yeah, I was going to say over 1 million. For
2.5 million base pairs.
Oh,
wow.
Is that right?
Wow.
Yeah.
Rich, what's the biggest pox?
About 300 kb, about this size.
About this size?
Mm-hmm.
Yeah, fowl pox gets up there around this size. Most of them are down around 200 kb.
And does everybody know where New Caledonia is?
I had to look it up.
I had to look it up too. Yeah, I had heard of it before, but I didn't know why. And it's in the South Pacific. It's out near Vanuatu.
The real question is, what were they doing there?
Why did somebody sample a muddy puddle next to the Noumea airport? I do not know. Or Noumea.
So Jean-Michel told me, it may have been in the TWiV. He said he was at a meeting once, and it may have been there. He said, and at a break, he looked out the window and he saw a pond. So he dumped out his bottle of water and went and filled it. And maybe this is it. Maybe
Wow.
this is it. Maybe he was at a meeting in New Caledonia. Look, if it were me, I would have accidentally opened the bottle and drunk it on the plane. That would have been dumb. So this has got 452 open reading frames, potential proteins. They say it could be a circular genome, or maybe a terminally redundant linear circularly permuted molecule. Condit, do you know what that means? You know what that means?
Yeah. So what that means is that the way I think about it is in terms of its implications for replication. Some viruses replicate as the model is a rolling circle model, but the result is that you get what we call concatemers, and that is a whole bunch of genomes lined up end to end. And if they're lined up in the same orientation, and then when you package them, you could package a linear genome, but if when you cut up that concatemer to package it, if you cut it up so that you get more than a full genome's worth, you'll get a full genome's worth and then the beginning of that genome again on the next chunk. So that's terminally redundant. The sequences on one end are repeated on the other end. But then when you take the next chunk, okay, you're starting at a different place.
Right.
You're not starting at the same end. So you march down a little more than a genome of that, and you get another terminally redundant molecule, but the start and end places are different. But if you take them all and put them together, it kind of looks like a circle. So that's what they call circular permutation. So the implication is that you started off with a linear concatemer and cut out bigger than genome-sized pieces. It doesn't actually have to be circular, but the sequence looks like it's circular. Go ahead. Sorry.
They just mentioned that it's like a retrovirus, but when you were describing it, Rich, it reminded me of, aren't there some phages
T4.
that are like that?
T4.
Okay.
Absolutely. It's been around for a while.
Yep.
The rolling circle model can also be thought of as the toilet paper model of replication.
That's good. That's very good.
Because the DNA is just unrolling from there, and now we're gonna cut it up, but we're gonna get a little more than a square on. Yeah,
I have to remember that for teaching. That's perfect. Because they'll get that.
Yeah.
They do some sequence comparisons and find out that this is a new member of the Marseilleviridae family. And I just want to tell you the names of other Marseilleviridae. It includes Marseille virus, Lausanne virus, Tunis virus, Brazilian virus. Boy, that's a generic one.
Yeah.
And this is my favorite, the golden muscle Marseille virus. That just sounds so good, doesn't it?
Yes.
Golden muscle. I guess there's a golden muscle. And now we have a virus. And also, I like this one, Port Mu virus. So those are all Marseille viruses. Okay.
And New Caledonia is a French island, by the way. So these all because of the researchers who have led a lot of this work, these are all coming from French territories.
Now, one interesting observation that I think is pretty cool. So they want to know if there are any introns in this DNA genome, which would say it would have to go in the nucleus to be spliced and make mRNAs that would have introns in them that would have to be spliced because that happens in the nucleus. And they use a program called Exonerate.
Yes.
Because an exon, right?
Yes.
Is part of this, and introns and exon, and the program is called Exonerate. I love it. Don't you love it? All right.
I love it.
I'm sorry. You don't.
Yes.
It's okay.
It's okay.
No. It's good. It's good.
It's great.
So they don't really find any.
If the contig doesn't fit, you must acquit.
Ooh.
Oh.
They don't find any, but what they did find, so this is a hint that this virus may not be going into the nucleus, the genome. They do find some self-splicing introns, which is really cool. These are thought to be pretty ancient RNAs. These are RNAs that can splice themselves without the need of a spliceosome, a protein RNA structure. And I interpreted that as meaning that these are probably really old. So that's all I wanted to say about that.
No.
Before we get to the real cool part of this paper, I just want to mention, because probably Rich likes this, that the noumeaviruses, which are another family, terminate their mRNAs. So when mRNAs are made, they're capped, and then they're terminated and polydenylated. And it turns out that they in the noumeaviruses, there's a hairpin, an RNA structure, a base-paired RNA, a stem loop, if you want to call it, that is important, and that's where termination occurs. And they looked through the genome and found lots of potential such hairpins, and in fact, they showed that one of the transcripts, one of the mRNAs, actually terminates in one of these hairpins. So they follow the noumeavirus hairpin rule. What do you think,
Right.
Rich?
So I think that's cool. The notion of hairpins, RNA hairpins being involved in termination is, of course, not new. It's been around as one of the mechanisms as part of the catalysis of termination for quite a while. But it looks like it is pretty pronounced, pretty obvious in these viruses. It would be fun to look at, mechanistically. It implies that these viruses have a related mechanism of termination. It could be informative in terms of termination in general.
Yeah.
So it's pretty cool.
Mm-hmm.
All right. Now, the next thing they did was to compare the virion protein. So they purify noumeavirions, and they purify a closely Melbourne virus, another member of this family, and so these are purified virus particles. I think they do mass spectroscopy, right, to look at all proteins that are in the particle. So you can look at very small amounts of protein and find out exactly what they are. And they want to know what's in there. And that's where the surprises start to come up. They do find proteins in there of a variety. They have some cytoskeletal proteins, and some of them are shared between the two viruses. Some of them are unique. They find one protein which they say its detection in the particles suggests that nuclear components may infiltrate the factory in the cytoplasm where the virus particles are assembled.
The thing that I found interesting is that the most abundant protein is not the major capsid protein, but it's this unknown abundant protein. Is that the one? No, that's not the one necessarily that implicates nuclear leakage.
It could be that they actually suggest it. Yeah.
And then the second most abundant one is a histone H3-like protein.
Yeah.
That clearly suggests maybe something coming from the nucleus.
Yeah. So maybe the DNA is wrapped up in histones in the particle, and that's why that histone protein is there.
Yeah, maybe.
This one that you said is the most abundant, it's 150 amino acids, and that they come back to that.
Oh, yeah, okay.
It's a cool one.
I thought the histone-like protein that was so abundant was actually a virus-coded protein. Did I get that wrong?
It is right. It is a virus-coded histone, yeah. Yep. There's also virus-coded, these are host-encoded mitochondrial proteins and cytoskeletal proteins, so it's really picking up a lot of stuff in there. Now, there is a mRNA capping enzyme in the virus particles, which puts the 5' cap on mRNAs. There's also an RNase 3, which is an enzyme that's involved in the maturation of mRNAs, right, Rich?
Right.
Does POX have one of those?
Well, it's involved in the maturation of some RNAs in some places. No, I'd have to look back.
Okay, not important.
I don't think so. Actually, one of the classic, and maybe this has to do with their hairpin termination thing. One of the classic examples of RNase, matter of fact, where RNase 3, I think its discovery was wrapped up in the processing of bacteriophage T7 early messenger RNAs that are made as it's made as a 5-gene polycistronic thing with a hairpin in between each gene, and RNase 3 is a double-stranded RNA-specific endoribonuclease, and it recognizes those hairpins and cleaves them, and in the process generates 5 individual messenger RNAs.
All right. What they don't find, in which they say is a conspicuous absence, are any RNA polymerase, host or viral. And in other viruses, you can find polymerases and other proteins involved in the synthesis of mRNA. So I guess in a pox virus, Rich, you would find RNA polymerase in there, right?
Yeah, pox virus, you find RNA polymerase and the capping enzyme and methylation things and methylating enzymes and poly-A polymerase. So, in fact, you can purify pox viruses. You can purify the virions and feed them precursors for RNA synthesis, and the virions themselves will accurately make early messenger RNA and modify it at the 5' end by capping and methylating it, modify it at the 3' end by polyadenylating it, and actively extrude it from the virus particle so that basically it makes messenger RNA that's ready for translation on the cellular machinery. And there are, a number of, I guess it's mostly the pox viruses and a number of these giant viruses, apparently, though I'm not as familiar with them, that contain RNA polymerases, though I don't know that anybody's done the in vitro transcription experiment with them, which would be interesting to do.
Yes.
But there are other viruses, like Alan mentioned, herpes, adenovirus, polyomaviruses, papillomaviruses that do not contain RNA polymerases and presumably, or because of that, have to use the nucleus. They send their DNA to the nucleus. There is these viruses. There are DNA viruses that encode RNA polymerases that are not packaged in the virion, but they're RNA polymerases that are used late during infection. Okay, this includes both phage and animal viruses. In the phage world, the classic I've already mentioned is bacteriophage T7. It goes into the cell, and the first region is transcribed by a host RNA polymerase, and one of the gene products from that is a phage RNA polymerase, which then does the rest of the transcription. There are other phages that do the same thing. The animal virus that I think of that does that is baculovirus. Makes its own, actually quite unique. That's quite unique is not good English.
Just unique.
It makes a unique RNA polymerase that's transcribed. It's expressed early during infection and used late during infection for decoding the rest of the genome.
What you're saying is they go through a late phage.
Late phage, very good.
I couldn't resist. Now,
so these noumeaviruses appear to be replicating in the cytoplasm, but they're not bringing their own RNA polymerase with them.
That's the conundrum.
So then, right? How are they doing this?
And before we move on, I just want to make the point that they make the point that because they see this mRNA capping enzyme in the RNase 3, and those are seen in some of the other giant viruses in which the RNA polymerase is found in the virions, that's their argument that they're detecting these other things, and if they can detect these other things, if the polymerases were there, they would have detected them in the Numea and Melbourne viruses.
Right.
To me, one of the most interesting things about all this, I'll just mention it here, maybe we can come back to it later, is that not only is there no poly-A polymerase in the virion, but the virus doesn't even encode a poly-A polymerase.
That's right.
Okay, so you got to put a poly-A tail on messenger RNAs. That's got to happen. So unless this hairpin is somehow a different 3' end, you don't need a poly-A tail. I don't know. Okay, I don't know that anybody's ever looked to see if these RNAs are polyadenylated. But assuming that they are polyadenylated, it's not clear at all how that's going to happen.
Got it. So how would this happen? So one idea would be that the viral DNA could go into the nucleus, right? Or maybe something comes out of the nucleus to the cytoplasm to, as Alan would say, bootstrap mRNA synthesis. It could be an mRNA or it could be a protein that comes out of the nucleus that assists in getting those first transcripts of the noumeavirus made, and among those would be an RNA polymerase and everything you need to make mRNAs, and then it could do its own thing after that. All right. So they don't find any evidence for mRNAs encoding polymerases in the virus particle. They do find at 1 copy per virion an mRNA encoding a subunit of the RNA polymerase, but they think that's a bystander, they call it, right?
Right. Yeah. Actually, and I'd go along with that.
It's less than 1 copy per virus particle. Yeah.
Yeah. Forget it. That's worthless stuff.
Bystander noumeas. So then the experiment they do is quite interesting. They take some viral proteins that are known to be in either the nucleus or the nucleus. Now, these are from a mollivirus. One is a methyltransferase, one is a sumo protein. It has nothing to do with wrestling. And they attach GFP to these so that you can see them. And when these proteins are produced in cells, the green is in the nucleus or the nucleus. And then they infect them with noumeavirus. And at 1 hour, they diffuse out. They go into the cytoplasm. They have lovely pictures of that, right? Mm-hmm. So 1 hour after noumeavirus infection, these pre-labeled proteins, they can see them diffusing out into the cytoplasm. A lovely assay. You just have to look at it.
Yeah.
And the nucleus remains intact. They can stain it with a dye called DAPI that stains DNA, and it's still there in the nucleus. And then they start to see DNA staining.
So the nucleus is just kind of leaking out this protein.
Seems to be.
Right. That's my interpretation.
But it's remaining intact, but it's right. Losing some of this stuff.
Right. So and they have electron micrographs later on that show that the nucleus is all convoluted, not normal looking. It looks stressed out.
Oh, the word they use is just great. Circumvolutions.
Circumvolutions.
I don't know I've never heard of that. Anyway, So these proteins are leaking out. Of course, other things could leak out also, right?
Mm-hmm.
mRNAs
Right,
could leak out. We don't know.
Yeah, the largest of these is 62 kilodaltons.
Yeah.
I think that means with the GFP tag.
Right.
So we don't know if there's a size limit.
Yeah, right.
And the thing is that these are not proteins that, I mean, they shouldn't even be in the nucleus in the first place. Okay, these are I guess they're viral proteins from another virus that are in the nucleus.
Yeah, they have these. It's from a virus that are that presumably go in the nucleus during an infection.
But my sense is that if these can leak out, probably almost anything you could tag would leak out. I'm not convinced there's any specificity to this.
But I would have liked them to have used a cellular nuclear protein.
Yes, exactly.
Yeah, just to be sure, right, that nothing funny is going on. But within 2 hours, I think the leakage stops, and there's no more flowing out of the green fluorescent protein labeled proteins into the cytoplasm. So maybe by then, whatever is required for bootstrapping, the virus is there, and the virus is now making its own mRNAs.
And at 4 hours post-infection, here in the colored picture, as you say, it's now the proteins that were leaking out before are confined to the nucleus. And there doesn't seem to be, at that time at least, any significant green coloring to the viral factory. Mm-hmm. So at least these proteins are cleared from the factory. I have to assume that the stuff that leaked out got degraded. I don't think it went back in the nucleus. So there's no evidence one way or another.
Right.
But I would assume that it gets turned over. And so it's no longer in the cytoplasm, but still being made in the nucleus.
Right.
They do say that at 2 hours, later, just after 2 hours, the fluorescence outside of the nucleus is localized in the viral factory. It co-localizes with DNA, which they think is viral DNA. So that's interesting that it's localizing. Now, why would it localize there? This is a protein unrelated to noumeavirus, right?
Well, and it seems to me that there's a bit of a discrepancy here, because in the first picture it shows using GFP staining that it's pretty broadly diffused, and I don't. I don't
see the evidence for association with the factory.
Right. In the second picture, it's associated with the factory. Now, this could have something to do with intensity. Where maybe if you turn down the intensity on the first pictures, there would be foci in factories that would be revealed. I don't know, but to me there was a discrepancy between those two pictures. But at least
it's the next picture.
Figure 8, yeah.
It's figure 8 which shows that. Yeah.
Yeah.
They show viral nucleoids co-localizing with GFP in that picture. It's not figure 7, which you're looking at. It's figure 8.
Yeah. What I'm saying is there's a discrepancy between the two figures.
Yeah, I got it.
To me, because it's sort of a general diffusion in Figure 7, and in Figure 8, it looks like it's concentrated to the factories. However, there's no denying that in Figure 8 there does seem to be some sort of specific association with the factories. Your question is totally legitimate. This is relative to the virus replication. This is an irrelevant protein. So why would it be in the factories? I don't know.
It remains to be addressed. Okay. Well, that's the main observation here. And now one more thing that they do an electron microscopic study of the infectious cycle, which is quite nice, a series of photographs where they start by adding the virus particles to cells. They can see them being engulfed by the amoeba, brought into small vacuoles in the cells. And then they can see at this point the nuclei begin to lose their spherical appearance. And then they form circumvolutions about up to 30 minutes. And then the viral RNA, the virus begins to change as well in the vacuole. They believe that the capsids are dissolved and the core is transferred to the cytoplasm through an opening in the membrane, the vacuole membrane. And then the nuclei return to normal after a while. I think it's 2 hours or so. And then new virions, listen, this is very interesting. New virions are first assembled quite early on, I think. couple of hours after infection. Is that what I'm seeing here? Would that make sense, Rich?
I would expect it to take longer than that. I think those early ones they're talking about when they got the arrows in those figures, I think those are infecting particles. And I think it takes a lot. This factory, I'd have to blow that way up. But later on, I think it's going to take much longer, at least 4 hours, and later than that to get
New
maturation to begin.
They do say that at 7 hours, they have particles in newly synthesized virions in vacuoles, which then they say these may exit by exocytosis. So 7 hours, they're done.
Right.
Or at least the first ones are done. So
I looked up circumvolution.
Yeah.
It's a winding movement, especially of one thing around another.
I'll be darned.
Huh? You know, Jean-Michel and Chantal, they have wonderful words that they use. In fact, Alan, bootstrap is in this paper.
Right.
They do use the word. I know he likes to use that. So there's an interesting discussion. Let's touch on some of the topics, and then we'll give a summary again. And they believe that this could be a transitional evolutionary stage between the viruses that are purely cytoplasmic and those that are nucleocytoplasmic, that have a nuclear phase. They require the nucleus, right? Yes, Rich.
Yeah, I want to make sure that we haven't really described specifically what the implications of this are. What we've done.
That's right.
We focused on the facts. The facts are that the virion doesn't contain an RNA polymerase. The fact is that the replication appears to happen in the cytoplasm, and I have something to say about that as we go on. And the other fact is that the nuclei get leaky. The implication, which to me is not proven, is that among the proteins that leak out of the nucleus will be transcription enzymes, right? That go to the DNA that's in the cytoplasm and transcribe the early genes of the virus to bootstrap the infection. That launches the infection. Once you've done that, you're golden. You can make new viral RNA polymerase. You can make DNA polymerase, and go along with the.
You can make anything that's encoded by the viral genome. Yeah.
So the implication is that RNA polymerase is exported from the nucleus to the cytoplasm to the factory to initiate the infection. But that concept, that idea, is not proven. Right? They didn't look at RNA polymerase. They didn't show that early transcription happens in the cytoplasm, et cetera.
Do we know that the RNA polymerase and associated proteins is an early transcript at all?
No.
It would have to be to be correct, this idea to be correct, right?
Yes. Well, and in pox viruses, the RNA polymerase, the core RNA polymerase, is transcribed throughout infection. Okay. The stuff that's transcribed late encodes enzyme that gets packaged into virions, and the stuff that's transcribed early, it's sort of phase shifted. The stuff that's transcribed early is used late during infection. In this particular case, if you put the whole model together, the presumption would be that the host enzyme is exported from the nucleus to the cytoplasm, transcribes early genes, and that the RNA polymerase that is encoded by this virus would be among those early gene products. But as you point out, that's not proven.
So they need to track the host RNA polymerase either by labeling it with GFP or using an antibody and see if, in fact, it comes out into the cytoplasm early in infection and co-localizes with the viral genome, right?
Absolutely. And this kind of stuff has gone on with pox viruses for decades, where there's so pox viruses, which genuinely do, there's no question, but they replicate in the cytoplasm, they carry their own RNA polymerase, et cetera. Nevertheless, there is a requirement for the nucleus. Okay. If you inactivate the nucleus with UV, or if you actually remove the nucleus from the cell, which is a really interesting trick, the virus can do all of its gene expression, but it can't put particles together. And people have wondered over time whether, what the nature of this is. And this has resulted in studies that suggest that host enzymes including there have been reports that host RNA polymerase and host transcription factors come out of the nucleus and go to factories and assist in the virus replication. But some of those studies were flawed, and nobody's ever been able to prove absolutely that any of these transcription factors or enzymes actually contribute to transcription in the virus infection. Those are hard experiments to do. That's a hard proof to come by.
Right.
One of the problems with the RNA polymerase experiments is that most of these RNA polymerases have a lot of homology. The poxvirus RNA polymerase has homology to the host cell RNA polymerase, so that there could be cross-reactivity with antibodies. So if you want to use antibodies to track these enzymes, you could run into trouble. But now with the more modern techniques like tagging and GFP labeling, you could probably distinguish. You should be able to distinguish between the host and the viral RNA polymerase and do those experiments. Whether you can do it in amoeba or not, I don't know.
The other question is, what in the virus particle, and it has to be something in the particle, is making the nucleus permeable so that things come out.
Absolutely.
Right?
Absolutely.
So it has to be brought in, because you can't do any transcription. You have to have a protein in the particle, and it may be this abundant 150 amino acid protein, right? Which makes sense. An abundant protein comes out. So I guess they're making antibodies to that and seeing if it's going in the nucleus, right?
So whatever causes that has to be transient, because then the nucleus comes back together again and looks okay late in infection. So it's something that the virus is bringing in with its particle, and that small protein is a good candidate.
How it works is really interesting, right? If it specifically affects the pores, or it does something. It sounds like it's doing something global to the nucleus, right? Circumvoluting it.
Right. And then if it is made so that it gets packaged in the virion, then at late times something has to happen so that it's not making the nucleus leaky again, potentially.
That's right. Yep.
So I want to bring in one more sort of caveat, which they don't discuss here, that I think is really important, and that is that there is another fundamental mode of DNA virus replication that they have not discussed in this paper, and that is iridoviruses. And iridoviruses, these are invertebrate. Actually, they infect vertebrates as well. There's insect. There are double-stranded DNA containing viruses, fairly large, as we already discussed, circularly permuted, terminally redundant genome, et cetera. These include the ranaviruses that are trashing the amphibians around the U.S. The sort of laboratory prototype is frog virus 3, which has been studied extensively, but there are also insect iridoviruses and et cetera. They have a very peculiar way of replicating where now they encode an RNA polymerase. They do not package it in the virion. The DNA goes into the cell, and the DNA actually enters the nucleus and goes through a preliminary round of replication in the nucleus, and early transcription happens in the nucleus, and then the DNA is exported to the cytoplasm. And the rest of DNA replication, what I would call vegetative DNA replication, the really robust stuff and the rolling circle stuff, happens in the cytoplasm, where you get the appearance of a viral factory. And late transcription happens somewhere coincident and dependent on DNA replication, presumably in the factory, though that's not been proven. And morphogenesis happens in the factory. So in the case of iridoviruses, the DNA sort of sneaks into the nucleus for a little while to get launched, and then splits and goes to the cytoplasm. And I don't think that there's enough study of this particular virus to eliminate that as a possibility.
I agree.
Okay.
I agree.
Yeah,
I agree.
So back to the bench, guys.
I'm sure they're at it. I'm sure they are doing all sorts of things. Yeah.
Yeah. So I want to see whether as we've already said, whether the RNA polymerase comes out and specifically goes to the factories. I want to see convincing proof that the DNA doesn't enter the nucleus. Probably one of the ways to do this is to do some sort of in situ hybridization with something that you know is an authentic early message, and see where that's produced. Is that actually produced out in a factory or in a nucleoid, or is that produced in a nucleus? So where does early transcription occur? I think that's a critical experiment.
And so, yes, it could be that some DNA is actually getting in to the nucleus, and because as you said, they haven't looked at it here.
Right. And that's something you wouldn't necessarily see just by a microscopy experiment.
No, not at all.
You're going to have to do that by, either some sort of hybridization experiment where you specifically light up the small amounts of viral DNA, or a partitioning experiment or something like that. But I think
put in a labeled DNA to start with, a virion that has.
Yeah. Mm-hmm. You could do that. I'd like to see where early transcription happens, but I don't even know if they have necessarily enough molecular biology on these things to say that such and such a transcript is absolutely an early gene.
Right.
Okay. I would think RNA polymerase would be an early gene.
Should be.
You could do that. And then there's the remaining question of the poly-A polymerase. That mystifies me. And I just sort of made it up as we were discussing it, but I wonder if they've actually ever looked to see if these RNAs are polyadenylated. And maybe they aren't. Maybe that hairpin substitutes somehow for a polyadenylation. So lots to do.
Right. All right. One more thing. The evolution of this kind of virus, as I started to say before we finished talking about some important things. So this kind of, if all this is correct, that this virus pulls something out of the nucleus that it needs to make early transcripts in the cytoplasm, this would be kind of an intermediate between viruses that go in the nucleus and those that stay in the cytoplasm, and that is what they say, an intermediate level of viral dependency on the host. Now, in terms of evolution, this is an interesting discussion. As we've talked about before, there are two sort of broad ideas of the evolution of eukaryotic viruses. One is that viruses started out big and got smaller, so they arose from a cell with lots of genes, and then they lost them, or viruses started out small and their genomes expanded. The genome expanded hypothesis. And their thought is that genome expansion allows you to get enough genes that you don't need to go in the nucleus anymore if you're a DNA virus. Right?
These are not mutually exclusive hypotheses.
No, but there are some people who think they are.
Yes.
Like Eugene Koonin doesn't believe one of the two, and I don't remember. Does anyone remember?
Eugene, Eugene doesn't. Eugene's into the acquisition model.
Yes.
And this all goes back to this thing about the fourth domain of life, because the fourth domain of life is a model that favors the I was a cell and I lost stuff and now I'm a virus. Okay, and that's that bit is we got viruses that have a bunch of eukaryotic looking genes in them that initially, when they were discovered, was thought that they aren't genes that are in any of the existing domains of life. So there was a fourth domain of life from which this virus is made, and that fourth domain of life is now gone away. But Eugene, in looking at that, says, no. In fact, you can find homologs for all of these genes in existing life forms. So you don't need the fourth domain of life theory. Right. That doesn't necessarily disprove the cell to virus thing, but Eugene favors the other one.
Do we do that paper on TWiV?
Yeah, we did. Just a couple of. There's been a couple of papers, but just a couple of episodes ago, we did that one.
So as you're right, Alan, you could have both going on here, right? And they're both.
There's no reason why we have to have viruses having a single origin.
No, and either way, it's going to be very difficult to sort out which is happening. But you can imagine with either scenario, whether you start out small and get big, or start out big and get small, you could have an intermediate where the extremes are. The viruses are in the cytoplasm. They have everything they need to make mRNA to replicate their genomes and make new particles in the cytoplasm. And then as they lose genes, they get more and more dependent on the nucleus, and maybe this virus is an intermediate. And then finally, they've lost everything, polymerases of all sorts. Now they have to put their DNA in the nucleus. And they say this golden muscle Marseille virus has a frame-shifted RNA polymerase gene in it, which they say could be a step along the progressive loss of function heading towards the nucleus. right?
Or it could be a step along the progressive gain of function.
Yeah, it could be either way. It could be gain or loss, for sure. You can imagine this as an intermediate between the two. If you start out big, you're in the cytoplasm, and you lose genes, you have to go in the nucleus. If you start out small, you start in the nucleus because you don't have any polymerases. You gain genes, and you move out. And maybe the last step before being totally independent in the cytoplasm is this noumeavirus, where you're mostly in the cytoplasm, but the bootstrap you have to dip into the nucleus. Anyway, that's a cool. I like thinking about that because this is old stuff that happened many years ago, and it's fun.
Yeah, it was TWiV 437, the giant virus with many translation system components.
That was a Kunin paper, right?
Yeah, well, Kunin was on it.
He was on it.
And he did all the probably computational biology, because I'm familiar with his diagrams that he does. Computational biology that said that all these translation system components in this giant virus relatives can be found in existing life forms.
All right. Who would like to summarize? Don't all crickets, man. Crickets.
Okay. So let me try.
Go ahead. Rich, you should, because it's a giant virus, and that's what you are.
So this paper really explores fundamental mechanisms of DNA virus replication, where the existing dogma, if you like, is that there are two fundamental mechanisms. One is the virus takes its DNA to the nucleus and uses nuclear functions to replicate. The other is it does it entirely in the cytoplasm. What's really novel about this paper is that this is a virus that replicates apparently by what you can see in a microscope in the cytoplasm, and yet doesn't have all the equipment that would be required to do that. And the paper suggests that equipment is actually exported from the nucleus to the cytoplasm to help the virus complete its replication cycle. And if that's true, this is a totally novel mechanism that is intrinsically interesting and also has implications for evolution of viruses.
Yeah. Right. And we're excited because this is something new. The idea that a virus to bootstrap in the cytoplasm is pulling something out of the nucleus. And that's where they say in the title, remote control of the host nucleus. Do you think it has a lot of buttons, this remote control?
Nuclear buttons.
Yeah.
Do they need a code, Alan, before they can push them?
Gosh, I hope so.
One thing I just would like to add to that summary is that the way that they know that this is doing things differently is that the purified virions don't seem to have the transcriptional machinery that they would have if they completely did their thing in the cytoplasm, like other cytoplasmic DNA viruses.
Right. Let's do a couple of emails. The first one is from our friend in Oregon, Ken Stedman. Dear TWiV fellow plaque lovers. Just finished listening to TWiV 437 while, you guessed it, doing a plaque assay. Only 5 plates, not 200. My wall will be pretty small. Wonderful, and not at all boring. TWiV on viruses that don't make you sick. I must admit to an inordinate fondness for spindle-shaped viruses. Maybe I will have to compete with Kathy. Shameless plug. See the cover of the next issue of J Virology, Volume 91, Issue 10. But who looks at journal covers anymore?
That's why I pasted it in for us.
Yeah, there it is. That's beautiful.
That's cool.
Neat.
This is his spindle, the sulfobolus spindle-shaped virus 1. And, he has a belt buckle in that shape also. Mm-hmm. And if you follow me on Instagram, instagram.com/profvrr, you will see from ASV a couple of years ago a picture of it. Yeah, it's very pretty. Resuming his email, the bicauda viruses that were found in the cork samples, that's the paper we talked about 2 episodes ago. Those are those cool things on the ocean floor, right? Cork from just off the Oregon coast are fascinating. One in particular, ATV, is known to undergo an extracellular morphological change in the virion whereby it grows tails. Whether the subsurface ones do the same is an open question. This is a question from a subsequent email, which we may not get to, but Hopefully I'll remember next time. Thanks for not one, but 2 TWiV bumps. Hope that they will help with my 3 pending grant proposals. I was also very pleased to hear Kim's email about the CPhages program at Washington State University, run by a buddy of mine, William Davis, a great program. I'm trying a somewhat similar course-based undergraduate research experience, a.k.a. CURE, Mutant Viruses from Hell, Advanced Molecular Biology and Cell Biology Research Laboratory, some of whose results are in the aforementioned JV paper, which he provides a link to. Sorry that I will miss the DC March for Science and the live TWiV, but will be participating in the Portland March. Hope that you will have a grand old time. Weather in Portland, 14C, classic April weather, either pouring with rain or bright sunshine. Wait 5 minutes and the weather will change. For Vincent, the cherry trees are just starting to bloom. I also have a listener pick, which I don't think TWiV has covered yet. The Fab Lab with Crazy Aunt Lindsay, a YouTube channel with kids do-it-yourself science projects from making butter, citrus batteries to yeast growth, etc. It's a lot of fun and definitely another science communication channel. Provides a link. Second shameless plug. Stay tuned for a guest appearance on the Fab Lab by Extreme Virus Prof in the next season. And that's him, of course. Ken. Thank you, Ken.
So I looked at the Fab Lab. It's nice. It's nicely done. I laud this woman for doing what she's doing. A lot of kids' science experiments. Little kids.
Yep.
Very cool.
Alan, you're next.
Sure. Jessica writes, Dear Dr. Racaniello, I'm an admirer and longtime listener. Thank you for your podcast. I'm really happy you're marching as one of my favorite scientists and educators. That said, I'm also disappointed that many scientists, including on the TWiV team, did not join the Women's March to promote the science and fact agenda, as I and many others, Dr. Spindler, did. Sure, it was mentioned on the show, but felt more like an observation or musing than anything. This lack of support and participation in January feels to me like a perpetuation of the bias against women that remains pretty dominant in the STEM fields. Our male counterparts are comfortably outspoken about the usefulness and criticality of science and data, but not about equality. I feel like it's an affront to female scientists that our male colleagues were not as motivated to march for social justice as a cause that may directly impact funding and jobs for them personally. I'm still a fan, and I'm glad you're participating in the resistance and for a great cause, but I'm just sad at the fact that you missed an opportunity to buoy a movement that, whether obvious or not, impacts you, science, research, and society at large. I just wanted to let you know, as someone who has spent the majority of my life studying and working in science as a female, that I will not march because I did already, and I promoted science back then as I was encouraged to do. There were actually many signs about facts, science, and climate change, to name just a few, so I was not alone. Good luck on Saturday. And Vincent, you wrote to her, and...
So I did write, and I said, you're absolutely right. I should have gone. I feel very badly because my wife and daughter went. I could have just gone with them, and I don't know. These points are great. So I wrote basically to apologize, and then she wrote back. So go ahead.
So she wrote back, I was really conflicted about writing you, as I've been really conflicted about whether or not to participate in the march. I really do admire what you've done with TWiX in educating listeners worldwide. I admire that you defend research and the scientific method, including by marching for science, but I wanted to let you know that I had this internal struggle. I can relate to your personal feelings of disappointment because when I participated in the Women's March in January and read many opinion pieces from women and people of color citing their disapproval of the march, it enabled me to understand that dismissive sentiment is valid and fair. I hadn't thought about it at the time I signed up for the march, that I had been absent, quote unquote, from many important demonstrations before the Women's March. We all could have, should have, marched many years ago to insist on justice and equality for our friends of color, women, refugees, immigrants, etc. In a recent example, we, marchers, should have showed up when the Black Lives Matter movement mobilized, but many of us didn't, and we failed to drive the necessary change. We all have blind spots, it's part of life, but those of us who have privilege can change the world if we recognize those shortcomings and make a point of helping where we're needed going forward. I'm committed to showing up and demonstrating for justice for all as often as possible. I really appreciate your response and the efforts you've made previously to promote more diverse representation in science. I also sincerely appreciate that you'll use your influential position to continue to lift up your underrepresented colleagues. Thank you so much again for your response. I really do look forward to hearing about the March and great new episodes of TWiV. I promote the podcast often as a biosafety professional supporting R&D because it's important to me that folks working in this field understand the science.
I love the part where she wrote, those of us who have privilege can change the world if we recognize these shortcomings. I think that's great. I love it.
It's an excellent point. And I would just add that it is hard to identify your own privilege because the nature of privilege is that it hides itself from those who are experiencing it. It's obvious to those who do not experience it, but it's virtually invisible from the inside. So you have to make a conscious effort, and I do try, but I know that this is something that is going to perpetually be a blind spot.
Yeah. Kathy, you have any thoughts?
Two things. While you were reading, I went back to look at my pictures from the Women's March here in Ann Arbor, and if it's not 50% men, it's pretty darn close in most of the pictures that I'm looking at.
Good.
And just a fun kind of fact about privilege. So the GI Bill came into effect after World War II. Is that correct? I'm not sure of my history.
I think that's correct.
I think that's the case. And that actually is a form of white privilege, because how many GIs were white versus how many GIs were non-white?
Yeah.
It's just that kind of thing. Like Alan was saying, you don't recognize it because it's not something that proclaims itself.
Yeah.
Right. And yet, at the same time, the military has, in many other areas, been at the forefront of desegregation.
Mm-hmm.
But yeah, things that seem to be benefiting everybody are not necessarily benefiting everybody equally, just because of the structural discrimination that is baked into our system from its origin.
Anyway, thanks, Allison. Appreciate it.
Well, just for the record, I was at the Women's March in Austin, along with my wife and daughter, and it was a blast. Everybody turned out. It was a beautiful sunny day. It was my first taste of Austin in action, and it was really heartening. It was really very cool. You know what the local motto is here? Keep Austin weird.
Right.
as promoted by the Austin businesses, and they live up to it. It's terrific. I'm looking for a picture which I will send you of us at the march.
Kathy, can you take the next?
I kind of lost my place. This is Brian.
Brian.
Okay. Hey, docs. Love the show. I listen often to all the Took series, though TWiP and TWIM get more of my time. However, I wouldn't have discovered them had it not been for this show. I just couldn't let it go when Alan, who I normally would think has more sense and credibility to his statement, or rather misstatements, when he was talking about the size of the U.S. military. We are not the largest. That would be China with 2.3 million, which is about 750,000 more troops than the U.S., in second place with 1.5 million troops. Then you had to go a step further and say larger than the next 10 biggest. Well, that number of the next 10 largest, not the 10 largest, is 7,053,201 to be larger. Also, shame on Dickson, my favorite voice on the show, for affirming in the background with correct. I promise I forgive you both. I'm sure I'm not the only one to catch this, at least I hope I'm not, but I felt the need to make the correction. I don't mean to sound critical, but rather constructive. I know you are passionate about the issue at hand, the NIH budget, and probably didn't think that one through. Happens to us all. I am sure if my words were out there to be scrutinized by the masses, that I too would have several, if not many, misstatements. Anyway, I have beat that horse enough, and I am ready for another paper. P.S. Be nice to Dickson. Only the listeners get to pick on him. Haha, just kidding. I love the banter and chemistry of the whole show, of the whole crew. You all make the show what it is. There couldn't have been a better cast for Seinfeld, and there couldn't be better for TWiV either.
Yeah, that's cool.
To explain, what I meant by largest was largest in dollars, because we were talking about the NIH budget versus the defense budget. And somebody has helpfully pasted a graph in here that shows pretty much the point I was making, that we spend $596 billion a year on defense, And if you combine, is this the next 7 largest? 3,
Yes.
4, 5, 6 expenditures? It's $567 billion. So I was pretty close. We spend massively more on defense than anybody else in the world, even large numbers of countries combined, even the countries that you might consider as potential threats. And there's not much excuse for it except that we treat our military primarily as a welfare program, a welfare program primarily for the privileged, by the way.
Reading this graph for China, it looks like their budget is around $200 billion compared to the $596 billion for the U.S.
Right. So they have a lot. They have a lot of people, but they don't
get
have
paid less.
They have, I think, one aircraft carrier that's always in port, and they have nowhere near the number of jets and such. So, that was my point, was that we spend far more on defense than anybody else, and there's not really a good reason for it.
No. All right, one more, Rich.
Oh, man.
What's the matter?
I was looking for this. I found this photo of us at the march. I was listening. I really was.
Okay, you don't have to. I don't mind if you don't.
I'm cool. And by the way, I pasted in that graphic. Allison writes, Dear TWiV Masters, as Vincent spoke about the yellow fever vaccine in TWiV 430, he called anti-vaxxers morons because surely they would be against it. But all of you have it wrong. We don't care what adults do to themselves. We worry about the kids, like the baby getting 5 shots in the photo below with 0 follow-up studies for any harm done. Because vaccines are considered harmless and left out of every equation, thus the results of this great experiment continue to be ignored and no mysteries are solved. When I told the nurses at the clinic that the DPT they gave my son had paralyzed him for 3 days, they ran. So I never brought any of my 5 children to doctors again. Does anyone remember back in the 50s when there was only the DPT and OPV and the OPV was broken up? Today, babies are getting 25 doses by 6 months and, gee, that's funny, observation of a most bizarre reaction called autism has been begging for scrutiny. The bottom line is that the vaccine debate is a subject divided between the witnesses of adverse effects in their children like myself and those whom have not seen any. I was lucky because no one could say that something else paralyzed my son. In his case, it was the tetanus toxin in his DPT shot lacking antitoxin. Understand that many anti-vaxxers know what they saw, and that is why they hold their ground, like my friend whose 3-year-old had the immediate reaction of a seizure to the Hep B shot right there in the clinic and stopped talking. Thus, either vaccines are safe all the time or the risk isn't worth it, or the risk is worth it, with the latter being more honest and already proven to millions of parents for whom the risk was not worth it, and no medals were handed out. At what point is someone allowed to consider the downside to injecting too much, too soon, and too many times? How can nothing go wrong when common knowledge admits everyone will react differently to the same drugs and the same microbes? But I do want to thank Vincent for his brilliant comment on TWiV number 16 at 23:40 minutes when he exercised the scientific method by saying, I feel personally that there isn't scientific evidence that vaccines cause autism, but this is a very complicated thing. It may be in a certain child under certain conditions with a certain genetic background. It does something that we don't understand at the moment. We have limits as to what we can do. I agree that we have limits as to what we can do, especially when science can't find grants for negative research, nor can they publish any negative findings. So my question is this: as more vaccines are being added to the schedule for children, at what point does one ask, what could possibly go wrong? Do keep up the good work. I have listened to all your podcasts, every TWiV, TWIM, and TWIP, and have enjoyed them thoroughly. Best regards, Allison. P.S. By the way, polio vaccines do not cause autism.
Hold on. That's the next part is me, VR.
Yeah.
Sorry.
Yeah. Right.
I wrote no vaccines cause autism.
Right.
Sorry.
Fine. You should be happy to know that they're not on the list. And although the OPV can be problematic in children with weakened immune systems because they can't mount a proper immune defense in the 24-hour window of time before the virus reverts to virulence, Vincent says that's wrong. So we have a lot to discuss here. Still, yeah. I like the OPV because it's non-invasive, uses no adjuvants, and won't leave children on the spectrum. Meanwhile, the jury's still out on the IPV because the body hates to be injected with phenol. And she sent some links and et cetera to...
These are all of the standard anti-vax talking points that we are kind of sick of hearing, but thanks for providing them. The whole too much too soon thing is total hogwash. If you've ever looked at a baby crawling around on the floor and putting every damn thing in the house in their mouth, and child falling falling over when they're trying to learn to walk and scraping their knee, and consider the billions and billions of antigens that child is being exposed to through every possible mechanism, the exposure in vaccines is not even a drop in the bucket. These are just very specific injections in most cases that are going to stimulate immune responses against particular things. And this notion that somehow the risks have been ignored or systematically buried is flat out wrong. And people in the anti-vax community know that because they frequently cite something called the Vaccine Adverse Event Reporting System, VAERS, in making their arguments. The CDC has a tool through which people can report potential adverse events after vaccines. These data are mined constantly by people in public health who use them as a basis for it's very problematic data because these are just anecdotal reports, but they are at least collecting them and mining those data and using them to try to discover systematic problems. And there are shots, for example, DPT, that are known to be problematic. And those problems are, in fact, disclosed straight up front on the piece of paper that you should have been given in the pediatrician's office when your son was about to receive that shot. That's known to be a problematic vaccine, which is a major reason that it was replaced a few years ago with a new version, Tdap, and that is doesn't have the same side effects. It may not have the same efficacy either, but it's a better choice. So this discussion of risk and benefit of vaccines is ongoing and vigorous in the public health community, and I hate this repeated refrain and chorus from the anti-vax folks that, this is all being covered up, and they say that vaccines, have no risks. That's just absolutely not true.
And somebody cited the up to 3 times their body weight. That's not possible, even if the baby weighs only 5 pounds at birth.
Yeah, she wrote that babies get 25 doses within the first 6 months of life that are up to 3 times their body weight. That's just not possible.
No.
And IPV does not have phenol in it.
Right.
No.
And what I like to point out is that one of the times when children are receiving these vaccines, that time coincides with when autism is often first observed.
Yes.
Temporal coincidence does not mean causality. There's more ice cream sold in the summertime. There are more drownings in the summertime. We do not conclude from these 2 facts that eating ice cream causes you to drown. It's the same thing with vaccines and autism. They're coincident in time, but that doesn't mean causality.
Yeah. And
there have been numerous studies precipitated by the fraudulent report of the association between vaccines and autism that cannot find any support for that idea.
Right. And that report, incidentally, was created through the financing of attorneys who were hoping to generate lawsuits against pharma companies. This is all extremely well documented in investigative journalism by a fellow named Brian Deere. And if you'd like to read more about it, Allison, another good summary is a book called Autism's False Prophets by Paul Offit, you probably have a picture of him on your dartboard, but you might want to give his book a try anyway because it's quite good. I do want to say that I am sorry to hear about your son getting paralyzed after the DPT shot. That could have been an adverse reaction. It could also have been caused by something entirely different that was coincident with that. In any case, it is generally recommended that if there is something believed to have been an adverse reaction to that particular shot, especially, because as I say, it's known to be a problematic shot, that you should not get a second dose of it. So there's already a protocol in place. Okay, this kid had a bad reaction to it. My wife, when she was a kid, she had a bad reaction to DPT. She never got a second shot of it. Now, she has subsequently gotten a Tdap, and that's been fine. So that is a known problem. It's very unfortunate when it happens, but if you have read anything about the three conditions that prevents, you should know that you were putting your other children at substantial risk by not getting them that shot.
I'm sorry that you've listened to all the podcasts and we have failed to convince you of the benefit of vaccines, Allison.
But at the same time, we're doing exactly what we've recently said is not the effective approach. We're sitting here telling you all the facts, which of course you have already heard. And I actually have a pick of the week that weighs in on this. So I know you're not going to go read Paul Offit's book because you believe Paul Offit is the devil. But I have a pick this week that I really do sincerely hope you go and look at. It's a cartoon. It's easy to flip through. It's non-threatening. It's the thesis of it is non-controversial, and I hope you at least give it some thought.
Well,
go ahead. It's interesting because we've talked about how in order to effectively communicate this kind of stuff, you need anecdotes. And I noticed that this letter is full of anecdotes of things apparently going wrong or going wrong associated with a vaccine or something like that. Unfortunately, you can't really come up with an anecdote that says, I gave my baby this shot and they got this vaccine and they got unsmallpox protected. Right. You can't, yes. The only thing you can do is show graphs and numbers. Okay.
I once heard somebody point out that we would not be having any of this anti-vaccine discussion if everybody who has been protected from a disease by a vaccine knew who they were.
Yeah.
You could not have these discussions because 50 people would pipe up and say, look, I didn't get polio because of this vaccine. You're not going to tell me this is a bad vaccine. And yet that's the case. That's what the numbers show.
You need to show a picture of somebody with smallpox and make the point that most people living in cities in the 18th century got this disease.
Yes.
All right. And now you don't.
Or
and
tetanus.
That's because of vaccines. Yeah, tetanus.
Yeah, do not want. But I will do as I often do in these situations and say my personal anecdote. When we adopted my daughter, she had immunization records from China, which we weren't entirely sure that those were correct. She had supposedly gotten all the shots she was supposed to get early on. We're pretty sure she got BCG, which is the one thing we wouldn't have gotten her, but because she has a little scar from that. But we looked at the little vaccination book and we said, well, do we really trust our child's life with these stamps in this book from another country? No. So we took her to the pediatrician, and after the initial exam, we said, we want to get her all of her vaccines as if she hasn't had any. Mm-hmm. And so she got, in one rather emotionally trying office visit that I had her with me, and I set my little baby on the table, and she was so adorable and trusting, and the nurses came in and they lined the needles up, and she was just lying there, and then they started like 6 shots, and she was just screaming her head off, and I felt absolutely awful. But I was reminding myself, this is what we do to protect the kid from these horrible diseases.
Yeah, well, that's the thing in the end, the kids.
And I feel like I know what I'm talking about, right?
Well, but that's not one of the arguments we're supposed to use,
Well, but it is in the sense that if this stuff is so dangerous, why would I expose my child to that?
Yeah.
Well, not just you, but millions and millions and hundreds and millions of people
Yeah.
have done it, right? The thing is that kids don't have a say in this, right? So
Right.
we have to make a decision to protect them, or in the case of people who choose not to vaccinate their kids, to leave them susceptible. And I think that's the real crime there.
But you
And by the way,
make decisions for your kids all the time when you put
Yes.
them in safety seats and seat belts and you feed them non-junk food. And yeah. That's part of being a
Well,
parent.
Alan, we're ready to hear your pick. What do you got?
Oh, yes. So let's go to picks. My pick is from a webcomic artist who goes under the name The Oatmeal. If you haven't already read his stuff, you really should. But this one is just particularly good. It's called "You're Not Going to Believe What I'm Going to Tell You." You're not going to believe what I'm about to tell you. And so just go to this page. If you want to show this to kids in a classroom, he's now released a classroom-friendly version. There's a little bit of foul language in this. And it's just a series of cute little cartoons leading into it with a couple little birds, and then you follow along, and he's going to tell you a couple of things. And initially you are going to be told things that you will easily believe, and then you're going to be told some things with citations that you may find a little harder to believe for reasons that he explains. And it's just a really good exercise, I think.
This made the rounds
really
this
is
week.
good.
This made the rounds this week, right? Is everywhere.
Yeah, in social media, for sure.
Yes. That's where I saw it, and everybody was tweeting this. I said, okay, fine, I'll go look at this. I do like this artist. And I said, this is really good.
Yeah, it's very good. It's very, very well written.
Yes.
Rich, what do you have for us?
So this is just a little tidbit that I found on social media, and it's a little video of a robot that was built by a high school student that solves a Rubik's Cube.
So cool.
And it's very cool. And I've watched this several times so that I could figure it out, and it's really great because the robot, it has a little manipulator that can flip the cube over and over and actually, turn the sections. And it's another part of it has a camera. So it starts off by flipping the cube so that at one point or another, each of the 6 sides is exposed to the top, and the camera scans the top. And I figure must be recording what colors are where.
Right.
And then what I find entertaining is that when it's done with that, it sits there for a while, and I figure it's thinking.
Yes.
And then it proceeds to go through and solve the cube. And so I thought that was pretty amazing, and particularly amazing because a high school student built this thing.
Yes.
That's really cool.
It's great. And this little video has had 32 million views. What I would give for 32 million views of a short video. Oh my gosh. That is amazing. It just goes to show you hit the right thing, it goes viral.
Yep.
Kathy, what do you have?
Well, coincidentally, I've been saving this pick for a while because of other show-relevant picks. This is about a Rubik's Cube at the University of Michigan. It was in some ways inspired by a cube sculpture that's been on campus since the 60s. It's a black cube on its corner, and it's between the president's house and the president's office. And so supposedly the university president spins the cube on the way into work and gets the university going.
Great.
It's bigger, way bigger than life size, and very popular thing to go see. And a couple of engineering students were in some lounge or something, and one of them was playing with the Rubik's Cube, and the other one thought about how cool it would be to make a large, manipulable Rubik's Cube. And so a senior engineering class worked on it 1 year, and it turns out that because of the friction involved and the force involved to move the cube, they couldn't use the same kind of mechanism that's in the small handheld Rubik's Cubes. So they had to engineer a solution for that. And they didn't finish making the cube, but they willed it to the next year class of students. One student stayed behind, didn't take her job that she'd gotten so that she could continue to work on the cube. But this thing has been finished, and there are several videos that I have here, plus a story with photos. The still photos don't quite do it justice that you can get from looking at the videos. It's the world's largest hand-solvable, mechanically operable, stationary version of a Rubik's Cube. And it's up on North Campus. I haven't made it up there yet to take a picture of it. It's just in one of the engineering buildings, and I plan to do that.
That's cool. That's really awesome. Yeah. Next time I come, if I'm ever there again, I have to check it out.
Yes, we will go do that for sure.
All right. I have 2 picks, which I've been doing lately. I'm sorry, but I just find things I want to share. First is a Nature article about finally the NIH is going to limit the amount of grant money that a scientist can receive. This has been talked about for a long time because the number is 10% of grant recipients get 40% of the NIH research money. And that just doesn't seem right. You should spread it around, because when you spread it around, you're going to get more innovative stuff, in my opinion, anyway. So they're going to have a scoring system, a point system. You get so many points for each kind of grant, and you're limited to 21 points. It's equivalent of 3 R01s. So I guess an R1 is going to give you 7 points, right?
Right. I found the chart. I think an R21 gives you 5. I can't remember any of the rest. Oh, some PL1s give you more than 7.
Good.
Depending on the role or how many cores you have or something. Yeah. P's and U's.
Well, don't have to worry about me. I'm way far below 21 points.
It'll be interesting to see how major research universities figure out to game this.
They'll game it, right? They have to.
Oh, yeah. What I'm thinking is going to happen is that they'll rename the postdoc position to something like adjunct investigator, and those people will then be obliged to apply for grants to a lab that they're not actually running.
Yeah, well, here you can't apply for certain kinds of grants unless you have a assistant professor or higher. It's like R1s. You have to be an assistant professor. You can't be another kind of appointment, but you can for R21s.
I'm sure there are ways to work with this, but it'll be interesting to see what those are, and to see. This is something that I think is a great idea.
Yes,
I think it's a wonderful concept, because there's ample data that these heavily funded, superstar labs are actually less productive than distributing the same amount of money to a number of smaller labs, depending on how you measure scientific productivity. But this is clearly a step in the right direction.
Yeah, I just hope it happens, and as you say, I hope they don't go around it, but they probably will, because we should spread that money out. Come on.
Yes.
My other article is an article by Paul Waldman in The Plum. And as everyone may know, this week the House voted on an alternative health care bill. And this article is called "Every Republican Who Voted for This Abomination Must Be Held Accountable." It has a very nice discussion of what was done. A lot of bullet points. Here are some of the things it does. Takes health insurance away from 24 million Americans, revokes the Affordable Care Act's expansion of Medicaid, et cetera, et cetera. I just read today, actually, in the Times, that since the Affordable Care Act was passed, personal bankruptcies have declined precipitously.
By half.
Isn't that amazing? Because people have to pay so much in healthcare money, they go bankrupt. Anyway, the real beauty of this is the second paragraph where he writes, "I won't mince words. The healthcare bill that the House passed this afternoon in an incredibly narrow 217 to 213 vote is not just wrong or misguided or problematic or foolish. It is an abomination. If there has been a piece of legislation in our lifetimes that boiled over with as much malice and indifference to human suffering, I can't recall what it might have been, and every member of the House who voted for it must be held accountable." Now, I know many of you don't like me talking about politics, but this is about health care, and this is a health and science podcast. And that's why I mention this, because everyone deserves good health care. And I don't think this is consistent with that. This is consistent with other things. The other things is the political part I will not touch, but
this is appalling.
Unbelievable.
Yeah. And it's opposed by an incredible assortment of organizations and individuals. Okay, the American Medical Association and the insurance companies. It's nobody likes it except those who voted for it. I'm not even sure they like it.
Yeah.
All right, we had two listener picks that we mentioned. One was from Ken Stedman, and let me go back. I have to go way up.
I pasted it down below.
Did you? Okay. So
the Fab Lab with Crazy Aunt Lindsay. It's a YouTube communication channel.
And then Maureen, we read her letter earlier. She said, I wrote a letter earlier about doing a summary of papers in your podcast, but forgot to add my pick of the week. And this is simple science experiments you can do with eggs before breakfast. This dad is amazing.
And it just leads to one video after another. They're very short and they're fabulous. So it's the eggs one, and then you eventually get to one that's a hovercraft on a CD or DVD. It's great.
One of the best parts about it is to watch the kids.
Yeah.
Okay. These are little kids, and the looks on their faces as they're watching us go on is great. One of my favorites is one of these double conical things that appears to roll uphill. Okay, and the kid is just delighted with this thing. It's great.
I like them because they're slightly sped up, so that gives them a quirky aspect.
Right.
And they're edited, so you don't have to wait too long for things to happen. So that's good. So thank you, Maureen. That's very cool. All right, that'll do it for TWiV 440. I kind of like the number 440. You like that? Does anyone?
Yeah. It's an A, right?
Yeah, it is. Yeah.
TWiV 440. You can find it at Apple Podcasts, microbe.tv/TWiV. And if you have a program or an app called Overcast or Downcast or Podcast, whatever you want, you can subscribe for free to podcasts.
Spincast.
Yeah, there are lots of them. Just go search TWiV, This Week in Virology, or the others. Just subscribe. So, you'll get them automatically, and you can listen or not. It's up to you, but we want you to subscribe. That's the key there. And if you have questions or comments, send them to twiv@microbe.tv. And consider supporting what we do. We have a page over at microbe.tv/contribute where you can find out the different ways you could support us, Patreon, PayPal, Amazon affiliates account, and so forth. And that's so that we can do more things, make more podcasts, travel a bit. Like we're going to go to ASV, all of us together, and so forth. So we went to the Washington TWiV before the Science March, etc. We can do those kinds of things and much more with your help. We really appreciate it. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
You're welcome. This was a lot of fun. And here's your A440.
Nice. How did you do that? You played a file on the computer? Or you have
a piano app on my phone.
Oh, you don't have a tuning fork, right?
No.
Rich Condit is an emeritus professor at the University of Florida in Gainesville. Thank you, Rich.
Sure enough, always a good time.
Alan Dove is at alandove.com. You can also find him on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. The introductory music that you hear at the very beginning of TWiV is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Not as viral as that 32 million video. Wow. That's impressive.
Yep.
Titles. Storming the Bastille. I hardly knew noumeavirus. I think that's great. I hardly knew noumeavirus. Surprising transcript reveals nuclear leak. That I get. Stealing nuclear launch codes. Hacking a nuclear launch. A muddy sample of fresh water. Nuclear circumvolutions. And the circumvolution is coming. So I like either I hardly knew noumeavirus or the circumvolution is coming. It's so great. What do other people like?
Well, I liked stealing the nuclear codes. I think that was it.
Stealing nuclear launch codes.
Stealing nuclear launch codes. But, I hardly knew me a virus. The others are great.
Mm-hmm.
What do you think, Kathy?
I like I hardly knew me a virus.
Right.
How about you, Alan?
That sounds good.
Yeah.
You always say it sounds good, Alan.
Well,
You make them
don't,
all.
yeah, I don't want to take a strong stand on one that I came up with. So
I hardly knew me a virus. I hardly knew me a virus. That's
Yeah.
great. I love it.